So it's a pleasure to be able to speak to you today. So the title of my talk is Human Epileptogenic Oscillations in Vitro, Old Friends and New Acquaintances. And just to give you an outline of the talk, what I'm going to try and do really is set the scene in terms of the advantage of using surgical tissue that's removed from patients who are undergoing elective neurosurgery for treatment of refractory epilepsy, and using that tissue for in vitro electrophysiological studies. And I'm going to show you some data that's already been published where we've identified oscillations which are translational across both the in vivo and in vitro point of view. And then I'm going to move on to demonstrate some novel anatomical data which we can correlate with these electrophysiological signals, and then finally wrap it up with some novel directions that we're now taking with this approach. So the first thing I wanted to do was introduce this chap, Norbert Weiner, and I like this quote from him which says, "The best material model for a cat is another cat, or preferably the same cat." And if you can remove cat and replace human, I think this is the kind of approach that we really need to be taking from a basic science point of view to understand human epilepsy. Now, in understanding epilepsy, why do we need to be using human material? Well, if we look at the armory that a neurologist has in terms of anticonvulsants that can be used for the treatment of patients that present with epilepsy, we've got a wide array of different types of drugs, but there is still this salient problem, which I think the field to some degree maybe is the elephant in the room, and this is that anti-epileptic drugs do not cure epilepsy. So 30% of patients are refractory to anti-epileptic drugs, and we have this issue in that we need to address this proportion of the patients that are drug refractory. So in order to do that, and using the ability to interrogate human brain tissue, we obviously need to identify translational biomarkers with which we can try and understand the underlying microcircuitry, and then use that to design new therapeutic interventions that can be used to treat this particular patient subgroup. And in thinking about a translational biomarker, well, of course, the likely candidate is the signal that underlies the EEG. So we know that the EEG is the gold standard in which clinicians such as neurologists and neurophysiologists apply to understand not only the type of seizure but the localization of the seizure. And this just shows the basic features of the epileptic EEG, where we have the interictal activity, which are these sharp wave discharges, which tend to be localized to a sort of broad area. So this is, I'm just using temporal lobe epilepsy as an example here. So this broad area is the orange portion. And then we have patterns which are associated with the ictal discharge, which inform us about the seizure onset zone, which probably lies within this broader irritative area. Now, one of the signals that has emerged recently, and this is really due to our ability to record directly from the brain using depth electrodes and subdural grids, and also the increased digitization rates that are now available, is the high frequency oscillation or very fast oscillation. And people such as Jean Gottman, who's based in Montreal, have really led the field in putting forward this idea that these high frequency oscillations are a very good biomarker that can be used for identifying parts of the brain that are involved in the epileptogenic process. So what we see here is that this is just an example to demonstrate that these high frequency oscillations or very fast oscillations are embedded within this interictal activity. So here's the raw unfiltered EEG, and you can then apply various types of band filters to pull out this activity. And this activity exists between 80 and 500 hertz and is sometimes split into two distinct groups, which are ripples, that is activity between 80 and 200 hertz, and then fast ripples, and that's activity between 200 and 500 hertz. And there is a debate in the field now about which is the best of these markers. Is it ripples or is it fast ripples? And perhaps that's something that we can discuss in the forum following this session. So not only is this pathological activity, high frequency pathological activity, associated with interictal discharges, but there is also numerous reports which demonstrate the presence of these persistent VFOs preceding ictal activity. So again, this is important because some people have alluded to the fact that this may be useful in terms of seizure prediction. But really, in trying to understand the clinical significance of these high-frequency oscillations, again, we must return to the work of Jean Gottman, who has published a number of very elegant papers where he's demonstrated that these interictal high-frequency discharges are an indicator of the seizure onset area independent of the classical ECOG spikes in the human epileptic brain. So he demonstrated that there was a high degree of both sensitivity and specificity associated with these oscillations between 80 and 500 hertz. So with this background in terms of setting the scene of this oscillation that may be very important in the process of epileptogenesis in the human brain, why did we think about using human brain samples to try and study this? Well, really, I was very much driven into this area by this excellent paper in which Professor Patachin is one of the co-authors. And this is where they tried to identify the research priorities in epilepsy for the next 10 years. And one of the things that they suggested was that we should try and identify seizure patterns and study the underlying mechanisms in human epilepsy using neurophysiological and functional imaging tools. And then secondly, try and reproduce patterns observed in humans in post-surgical human tissue. So with this, armed with this sort of call to arms with what we as a community should be really trying to focus on, we decided to use resected human epileptic neocortex to try and understand something better about epileptogenesis in the human brain. So why study epilepsy in resected human epileptic neocortex? Well, as I've already outlined in my introduction, pathological network oscillations are generated by local cortical microcircuits. And specific frequency bands, for example, these high-frequency oscillations, have distinct network mechanisms. However, it is worth bearing in mind that there is a huge degree of heterogeneity in terms of the microcircuits that are involved in the production of discrete frequency ranges. And at present, the current approaches such as subdural grids or depth electrodes do not have the degree of invasiveness that is required to assess mechanisms. Now, I'm going to be slightly provocative here and say that animal models have been productive, but there is this dilemma, which is that whilst they have provided lots of information, how much knowledge have we gained from them that has actually pushed the field forward and provided the cures that we are trying to identify? And there's a very nice review by Bob Slovater which really sort of encapsulates this idea of the information-knowledge dilemma. So what we've done in Newcastle is we've tried to utilise live human epileptic cortical tissue to assess the alterations in the neuronal microcircuitry that underlie this distinct form of pathological activity. So the first question is really, using this approach, are the patterns that I have introduced to you reproducible using ex vivo epileptic human tissue? So here is an example of some tissue. This is temporal neocortical tissue that we've obtained from patients who undergo the removal of the hippocampus for the treatment of their drug-resistant epilepsy. And the techniques that we use are predominantly extracellular local field potentials, which essentially are giving us a manifestation of the signal that underlies the EEG. And we can put in multiple extracellular electrodes to look at the location of various types of pathological activity. We can then post hoc stain the slice. This is just a Nissl stain, and then we get an idea of the location of where our recording electrodes were. Now, we've already published a number of papers in 2010 and more recently, in which we have clearly demonstrated the spontaneous presence of these high-frequency oscillations. So here are two data traces. We have an electrode which was the foramen ovale electrode, which was placed into the patient before surgery. And as you can see, you have the presence of these large interictal spikes, which when you filter them, you can see, I mean, it's even obvious from the raw data, the presence of a high-frequency oscillation here. And this is a recording from tissue obtained from the same patient, where again you can see the spontaneous interictal sharp wave with a very fast oscillation embedded within it. Now, this is a very important point in that we don't have to manipulate the environment in which the slice is sitting in vitro to observe these patterns of activity. So, similar to Richard Miles's group in Paris, this activity is spontaneously occurring. So we don't have to make the system more pro-convulsant by manipulating magnesium levels or potassium levels. So this is spontaneous epileptic activity that is arising from the local microcircuits in these slices that we have obtained from these human epilepsy patients. And because we can record this type of activity, these high-frequency oscillations, we can then go on to quantify the type of activity. And again, we see the types of peaks that have been observed in the in vivo recordings of these high-frequency oscillations in human patients. Now, it's also worth pointing out that in addition to observing interictal activity, we also observe spontaneous ictal discharges. I'm not going to talk about that today, but I do think it's worth pointing out. It's usually a comment that we get from referees that, oh, you don't see spontaneous ictal events, but we do, and that's future work that we're currently working on. So hopefully it's quite clear to you that we have spontaneous epileptiform activity that can be observed in this human surgical material. And what I now want to do is just share some of our experiences in terms of the data that we've collected from these recordings. The first thing to say is that, again, similar to in vivo recordings, we see the coexistence of these very fast oscillations associated with what we think are population synaptic events, and we see very fast oscillations occurring alone. So here on this side of the slide, you can see that this is our raw data that we've recorded, and what we've done is essentially filtered the data either to look at the slow sharp wave potentials or to look at the high frequency oscillations. And what we observed was that in fact there were two types of events which we simply classified type 1 and type 2. It was clear that you had high frequency oscillations which existed on their own or which were coincident with these large population synaptic events. And when we looked at the analysis of the peak frequency of these high frequency oscillations, there was no obvious difference. So these were both in what could be described as the fast ripple frequency domain. Now, of course, one of the advantages of our approach, which I alluded to in my introduction, is that we are afforded the manipulability that is not given to one when you're doing such studies in vivo. And this means that you can apply pharmacological agents to test specific hypotheses to gain a better understanding of the microcircuits that underlie this pathological activity. Now, there has been a large body of literature which has suggested that these high-frequency oscillations may be due to the involvement of GABAergic systems, and we wanted to test this hypothesis. So a simple way to test this theory is you can apply a blocker of the postsynaptic GABA A receptor, which is gabazine or bicuculin. And what you see here in panel A is the raw data traces at the top and then filtered to reveal high frequency oscillations. And what you should be able to discern from this is that in the presence of gabazine, you still see the occurrence of these high-frequency oscillations. This is probably better demonstrated in panel B, where we've got a slow time course recording, and you can see the wash-on of gabazine, and then we've plotted either the low-frequency events, which are the sharp waves, which you can see clearly get abolished as the gabazine washes into the chamber, whereas the high-frequency oscillations are still present. And so what this suggests is that spontaneous, very fast oscillations are preserved with reduced cortical GABA-A-mediated inhibition. Now we can take this a step even further, and combined with our local field potential recordings, we can record from individual neurons and then correlate their behaviour with how the network is behaving. So what we do is we use sharp microelectrode recordings to impale individual principal cells in this case, and we can then look at the contribution of either the excitatory postsynaptic potentials or the inhibitory postsynaptic potentials. And really the bottom line here is that the magnitude of synaptic excitation and inhibition onto principal cells is not correlated with the very fast oscillation amplitude. If you record from an inhibitory interneuron, and we know that this is an inhibitory interneuron because you can see that when you apply a depolarizing voltage step, you get this non-accommodating behavior. So this is the behavior of a classical fast spiking putative interneuron. And what you can see is that at the same time as you have this interictal sharp wave discharge with VFO, you see that the fast spiking interneuron only generates a single action potential. So you're not getting bursts of action potentials. And again, there has been this idea that is these bursts of action potentials from these fast spiking interneurons which underlies the high frequency oscillation. This is not the case from our recordings. However, when you look at the excitatory postsynaptic potentials that this fast spiking interneuron receives, you can see that there's clearly a bombardment and a very high frequency bombardment of complex and compound EPSPs, which when you correlate that against the high frequency oscillation in the local field potential recording, you see that the output lags the field very fast oscillation rhythm. Now, in addition, if you go back and look at your recordings from the principal cells and specifically focus in on the GABAergic IPSPs that they receive, you see that, again, most of this large sharp wave is generated by these large complex GABA-A mediated IPSPs. However, there is a slight hint of some high frequency oscillation down at the trough of this IPSP. And if you filter this event and correlate it with the local field potential, you see that there's no relationship. So again, all of this would really suggest that classical chemical synaptic transmission is not underlying the generation of the high frequency oscillation. So what is? Well, together with Roger Traub, we've been exploring the possibility that it is non-synaptic connections, that is, electrical connections formed by gap junctions, which most likely underlie the generation of these high-frequency oscillations. And in order to test that, we can do that pharmacologically by applying a gap junction blocker, carbonoxolone. And what you see here is that you abolish both the low frequency interictal sharp wave, but also the high frequency oscillation. More compounding evidence again comes from our intracellular experiments, where in a subclass of cells you can record these fast prepotentials or spikelets, which are thought to be a cellular correlate of electrical coupling between principal cells. And when you look at the relationship, the timing relationship between these spikelets and the high frequency oscillation, you see that there is a much more significant relationship. So the theory that we've put forward is that one of the underlying causes of neocortical epilepsy, and particularly the epilepsy that is seen in drug-resistant patients, is due to a pathologically excessive level of dysfunction in non-synaptic networks. In addition to describing this form of high-frequency oscillation, we've also more recently revealed a new form of high-frequency oscillation which has not been previously demonstrated in animal models, which we term the glissando. So these glissandi are brief runs in which the frequency of the high-frequency oscillation ramps up over time. And these are associated both with interictal and also with the ictal periods. And we've observed these not only at the level of the human epileptic brain slice, but also in human depth electrode recordings, where you can see that the glissandi precede the seizure. Now, I'm not going to go into these in any more detail. Rather, what I would like to now do is really, given the fact that I've introduced all of this electrophysiological data, I now want to tell you about some of the neuroanatomical work that we've been doing to try and correlate the changes that occur in the microcircuit and combine that with our electrophysiological recordings. Now, in order to do that, if you remember the first image where I showed you the dual local field potential electrode approach, really that's a very crude approach, and what we need is we need a much more higher resolution spatio-temporal technique which allows us to really sample as much information from the slice as possible. So our approach to do that has been to use the Utah array. This is a silicon array with 100 microelectrodes, 96 of those are live, with an inter-electrode distance of 400 microns. And what you should be able to see here is that this is our little human brain slice that sits in the recording chamber, and we simply bring this bed of nails down into the slice. So what you get is you get a slice. So this is a slice which we've done post hoc immuno on for non-phosphorylated neurofilament. Each of the red dots denotes where the electrode position has been, and the very powerful nature of this means that you can combine the electrophysiological recordings that you conduct. So, for example, this is a power map for high-frequency oscillations, which then can be overlaid on the neuroanatomical image. So you can co-register your electrophysiological data with a measurement of the anatomical networks that you're interested in. And so this allows you to correlate the neuroanatomy, and some of the things that I'm going to show in the next couple of slices include looking at specific markers for distinct types of inhibitory interneurons. We've also been doing some work with DI to look at the postsynaptic structure. And in addition, and Lydia's already nicely introduced this, we can resolve down to the level of the ultrastructure using electron microscopy. But what I want to do really to start off with this is to focus in on these interneurons. And we know now from the work of Yuri Buzaki and Peter Somogyi that network oscillations are sculpted by interneuronal diversity in the neocortex. So we have this very wide array of different types of interneurons classed on their neurochemistry, on their electrophysiology, on their postsynaptic targeting of the principal cells. And broadly speaking, they can be classified quite well on the basis of this neurochemistry, in particular the calcium binding proteins that they express. So we have the parvalbumin containing interneurons which target the perisomatic region. There's a subclass, the chandelier cells, which target the axon initial segment. We have CCK containing cells which target the perisomatic region, but in addition also target these perisomatic targeting PV interneurons. And then out on the dendrites we have calretinin and calbindin, and we also have somatostatin and NPY containing interneurons. So what we wanted to do was really address the changes that have occurred in these classes of interneuron and correlate that with our high density recordings in human epileptic brain slices. So here is two slices, one from an epileptic patient. So again, this is temporal neocortex from a patient with refractory epilepsy undergoing an amygdalohippocambectomy. And we also have what we term non-epileptic control temporal neocortex, which is obtained from patients who are having a deep tumour removed, so there's no epilepsy associated with their tumour, and the surgeons, in order to gain access to the tumour, have to remove the overlying cortex. So what we have here is both of these slices are stained for parvalbumin. In the non-epileptic control sample, you can see that there's a very nice uninterrupted band of parvalbumin staining here in the superficial layers. And just look at the epileptic sample. So you can see that you start off with a nice band of PV, and then all of a sudden the signal seems to drop out, and you get a reduction in parvalbumin expression. So if we overlay our Utah array recordings, and again, this is a power map in which the power map is for these high-frequency oscillations that are associated with the interictal discharges. You can see that where the PV signal starts to drop out, you see the emergence of this high-frequency oscillation. In contrast, in our control samples, it should be pointed out that we never see spontaneous epileptic patterns like I've previously shown in the earlier slides. And again, we can resolve down and look at these regions in more interest. So here is a chunk of tissue from the control sample. You can see lots of nice, healthy-looking parvalbumin stained interneurons with lots of terminals. And you can go down even further and see that these are clearly targeting the perisomatic region of the principal cells. However, in the epileptic sample, there's clearly a reduced density and also a significant lack of perisomatic targeting PV terminals. So really what I want to try and get across here is that there is a granularity of interictal activity which is correlated with a granularity in terms of the loss of parvalbumin signaling. And this has been previously reported by Javier de Philippe back in the 1980s, but they were using ECOG techniques to try and discern between spiking and non-spiking regions. And again, this is something that we can maybe talk about in the discussion about that approach. But really what I would like to suggest here is that in the regions where this parvalbumin signal is dropping out, we see the emergence of these aberrant patterns that are associated with epilepsy. Now, you're probably already thinking, well, he's only really looked at one subclass of inhibitory interneurons. There's all these other different classes that he described earlier on, and we have gone now and done a systematic analysis of these other calcium-binding protein-containing interneurons, using a very rigorous approach where we are able to go into these hotspots where we see the epileptic activity in our human brain slices and take images which then can be transferred to MATLAB and do analysis in terms of the density of these parvalbumin-containing interneurons. And so this is pooled data down at the bottom here. And what you can see is that there's clearly a significant reduction of the density of these parvalbumin-positive cells in the epileptic regions of these slices. We've looked at CCK, and interestingly, CCK, there seems to be significantly more CCK-positive cells in epileptic tissue, fewer perhaps in layer 6, but more in the superficial layers and also in layers 4 and 5. Calbindin, there are significantly more calbindin cells in the superficial layers of the epileptic tissue. And then finally calretinin, again we're seeing probably significantly fewer cells in the epileptic tissue, fewer in layer 1 and fewer in layer 4. We're also looking at somatostatin, and we have the data for that. It just hasn't been published, analysed yet. So really there is a diversity in terms of the alterations that occur in the interneuron population in the human epileptic neocortex. What we can say is that, generally speaking, the parvalbumin containing interneurons seem to be going down. We haven't been able to discern between the perisomatic targeting ones and the axon initial targeting parvalbumin interneurons, but that is work that we're currently trying to do. Clearly, CCK containing interneurons, the expression of those is up, and also we're seeing increases in calbindin, but decreases in calretinin. So there's some form of alteration that has occurred in response to the epileptic activity in these networks. As I already alluded to, we're using ultrastructural techniques to look at alterations in these hotspot regions of the human epileptic neocortex. And as Lydia has already introduced, we're able to discern between asymmetric and symmetric synapses. And our initial, and I would say that this is preliminary analysis, suggests that there is an increase in the asymmetric excitatory synapses and a subtle decrease in the inhibitory synapses. But again, we're using this technique hopefully to be able to discern between the axon initial targeting parvalbumin positive terminals as opposed to the perisomatic positive terminals. So really to conclude, and I hope that I'm staying to time, I hope what I've been able to demonstrate really is that pathological oscillations which are associated with epilepsy can be observed spontaneously in post-surgical samples that we obtain for our in vitro studies. We've been able to report previously seen EEG patterns, but also novel patterns that have not been associated with seizure generation before. The glissandi is one in point, which has not previously been observed in animal models of epilepsy. We feel that this in vitro approach offers unprecedented access and also manipulation of primary human pathological tissue. And the experimental techniques really allow an extrapolation between in vitro and in vivo neuronal dynamics. And ultimately, and I haven't really talked about this a lot today, but in combination with Roger Traub, our in vitro data helps to inform and refine in silico approaches. So what about future directions? Because really, what we really want to think about is how, as a community, are we going to provide cures, translational techniques that can provide cures for epilepsy. So here we have, this is at the top here, here's a very nice example of a green cell where clearly it's labeled for GFP, and we then stain for an antibody for CAM kinase 2 and then merge them, and you can see that there's clearly a nice merged image here. This is a more low resolution series of images that show that you do get multiple cells that are being infected with the GFP. And really the next step here is to obviously infect these human brain slices with viruses which have the opsin attached, and then to conduct optogenetic manipulation using this very cell-specific technique. Now the other thing to say is that this just, I thought it would be useful for any keen eyes in the audience that here is a recording that we've done of our interictal epileptiform activity pre-infection, and then following infection, so 6 hours later, this activity, when we put the slice back into the recording bath, is still sustained. So we feel that this is a very potentially powerful technique that will allow us to really identify the specific cell classes that would need to be targeted for optogenetic manipulation to bring about seizure suppression. So really what I want to say is that our approach in Newcastle is really to go from the level of the patient where we do ECOG, resolve it right down to the level of the seizure focus in vitro using electrophysiological and neuroanatomical techniques, and then finally in combination with computational modelers provide an in silico approach to this. And it leaves me just to thank all of the people who've been involved in this work, and various PhD students and postdocs in the lab, and of course my clinical colleagues who provide the samples and organisation of that. And just in case any of you might be in Newcastle next week, you're very welcome to come along to our Northeast Epilepsy Research Network meeting. So thank you very much for your attention.